Hi everyone. So in this video we will be studying about the muscle contractile proteins and the how the muscle contraction happens. Okay? So first we are studying about the muscle contractile proteins. In the contractile proteins we have the basically in the muscle we have the three types of proteins, the contractile ones, the regulatory ones and the structural ones. In the contractiles there are thin and thick filaments. The actin is thin filament and the myosin is thick filament. Then in the regulatory protein we have three proteins: troponin, tropomyosin and nebulin. Troponin is of three types. Troponin I for inhibition of interaction of the actin and myosin. So I is inhibiting the interaction between actin and myosin. C is binding site for the calcium, has binding site for the calcium, and T is binds the troponin to the tropomyosin. Okay, T is binding the troponin to the another T, tropomyosin. Then tropomyosin is binding the myosin binding sites. Tropomyosin is basically present on the myosin binding sites on actin proteins, and these are preventing the interaction. Then is nebulin. Nebulin is actin binding protein and regulate the thin filament length. Nebulin is basically maintaining the thin filament length. Then we have structural proteins. Structural protein we have actinin. Actinin is binding actin to Z line. Then is titin. Titin is binding thick filaments, that is myosin to Z disc and M line. The one which is binding actin to the Z line is actinin, and the one which is binding thick filaments, that is myosin to the Z line, is titin. And titin is basically the protein which is involved in the cardiomyopathy, dilated cardiomyopathy. Then desmin. Desmin is connecting the Z line to the plasma membrane. In the desmin, M is coming, so it is connecting the Z line to the plasma membrane. Then is dystrophin. Dystrophin provides scaffolding for the fibrils. Okay, so we are done with the proteins. Contractile is actin and myosin. Regulatory is troponin, tropomyosin and nebulin. And in the structure we have actinin, titin, desmin, dystrophin. Then another thing about actinin is it is of the spectrin gene superfamily and it composes dense bodies in the smooth muscle cells, cytoskeletal isoform along the microfilament bundles and adherence type junctions. So it is involved in dense bodies and in adherence type junctions. Then we need to remember that how the sarcomere is, what are the bands in that. Dark band is A band. Okay, the dark in the dark A is coming, so dark band is A band, and in between we will have light H band. We will be seeing it in the figure. Okay, light H band in the center and M line in center of it. So A band उसके बीच क्या है, H band उसके बीच में M line है. Light band जो हमारा वो है I band. In light I is coming, so light band is I band and it is divided by Z line. Now let's come to this. See, here dark band is A band, A band. Then we have I band, light band. In the A band we have the H band which is lighter, and in between the H band we have the M line. So remember AHM. AHM, okay, and then we have the I band, light band, and in between that we have the Z line. So which protein will be connecting the actin, that is thin filament, to the Z line, that is actinin. And these dark band is of the myosin filaments and thin filaments make the light band. Now let's understand the theory of the muscle contraction. So action potential opens the presynaptic voltage gated calcium channels. Okay, it will open presynaptic calcium channels. Then postsynaptic acetylcholine will be released, and then acetylcholine will act on the postsynaptic channels. Then after the acetylcholine binds and leads to muscle cell depolarization, and depolarization travels along the entire muscle cell and depolarization ultimately reaches the T tubules. Here T tubules, at the T tubules we have the dihydropyridine receptor, and it is just a voltage sensor. Okay, it is not a voltage gated channel, it is just a voltage sensor, and it will, and mechanically coupled to it is ryanodine receptor. Ryanodine receptor. So electrical and mechanical coupling between the dihydropyridine and the ryanodine receptor. Then the ryanodine receptor will cause the release of the calcium. What will ryanodine receptor do? It will cause the release of the calcium from the endoplasmic reticulum. So calcium will be released, and this calcium will be binding to troponin C. Okay, troponin C, and tropomyosin here is blocking the myosin binding site as calcium binds. जब calcium bind कर गया तो tropomyosin हट जाएगा, तो जो एक्टिन पे बाइंडिंग साइट्स है उस पे क्या बाइंड कर जाएगा? मायोसिन. एंड मायोसिन क्रॉस ब्रिज फॉर्मेशन विल अकर. Okay, myosin pulls on the thin filament. Then what happens? Okay, sarcomere, we know what bands we had. So which band will get shortened? H and I band, and between the Z lines, H and I band will get shortened. Okay, what is the mnemonic? Hi, I am wearing shorts. Okay, H, I and Z, these are contracting. The A band will remain of the, that is dark band will remain of the same length. Okay, then the muscle contraction has occurred. Now what will happen? How will the myosin head detach? Myosin head will detach with the binding of the new ATP. As new ATP binds, the myosin gets detached. And how will the muscle return into the muscle relaxation? Okay, muscle will relax after there is reuptake of the calcium. Reuptake of the calcium. The myosin head can bind to a new site on actin to form a cross bridge if the calcium remains available. Okay. So first, repeating again, first action potential comes presynaptic calcium channel, acetylcholine release, acetylcholine acting on postsynaptic channels. Then the action potential traveling to the T tubules, dihydropyridine, ryanodine. Ryanodine causes release of calcium from this sarcoplasmic reticulum. Calcium binding to troponin C. Troponin C causing detachment of tropomyosin. Myosin binding to actin. Myosin actin cross bridge formation. Then after the cross bridge formation, power stroke will happen and detachment of the myosin head when the ATP attaches, and relaxation of the muscle when the calcium returns to the sarcoplasmic reticulum. So this is all what we need to remember regarding the muscle contraction and regarding the muscle fibril structure. So I hope it will be easy now to remember about it. If you find my content useful, please do subscribe my channel and share my video. Thank you.